Greetings, students. Welcome to the second lecture on the histology of the respiratory system. And we start with the trachea. The wall of the trachea is formed of 4 layers. Starting from the inside, these are the mucosa. Mucosa is formed of a typical respiratory epithelium with underlying lamina propria, which contains also glands. Then we have submucosa. Submucosa. This is a connective tissue layer that contains some lymphatic nodules and mucous glands. There is a layer of elastic fibers that separates the lamina propria from the submucosa. And then the third layer of the trachea is the cartilage layer. The wall of the trachea is reinforced by about 20 C-shaped hyaline cartilages. Now, the C-shaped cartilages, of course, has a gap, and this gap is directed posteriorly towards the esophagus. And the gap, this direction of the gap posteriorly, so that the esophagus is allowed to expand. But the gap is bridged by some elastic fibers and a smooth muscle called the trachealis muscle. So this gap is not empty, but it's filled with elastic fibers and trachealis muscle. Finally, the outermost layer of the trachea is adventitia, which is a connective tissue layer, of course, that binds the trachea to the other organs. Here we can see in this image, this is the mucosa, epithelium and lamina propria. Here is the mucosa, epithelium and lamina propria. And then we have the submucosa containing the glands. These large glands are all contained within the submucosa. Remember, between the lamina propria and the submucosa, I have a layer of elastic fibers, a layer of elastic fibers, but because elastic fibers require special staining, I do not usually see them. And then the third layer is a layer of cartilage. However, posteriorly, the third layer is a layer of smooth muscles. And finally, the outermost layer is connective tissue adventitia. Then the trachea will divide to form the bronchial tree. First, we'll have the main bronchi, then secondary bronchi, and then tertiary bronchi, and we will have continuous branching in the bronchial tree to get smaller and smaller bronchi. Now, as the division continues, as the bronchi become smaller, several changes in the histology will occur. So the wall of the bronchi has the following layers: mucosa, smooth muscles, submucosa, cartilage, and adventitia. The mucosa is respiratory epithelium with underlying lamina propria. Lamina propria is ordinary, just like everywhere else we talked about, with some mucous glands and some lymphatic nodules. Elastic fibers are also present in this lamina propria. Here we have a layer of smooth muscles, immediately underneath the mucosa. Now, in the bronchi, the smooth muscles here are arranged helically along the bronchus, and they crisscross each other. So they spiral around the bronchus and they crisscross each other. Because I have smooth muscles, the epithelial lining is folded. The mucosa appears folded because of these muscles. The submucosa, connective tissue layer with several glands, and then cartilage. Now, remember, in the trachea, we had C-shaped cartilages with smooth muscles only on the posterior aspect. In the larger bronchi, the main bronchi, the bronchi that are found outside the lung, the cartilages are ring-shaped. And we have more smooth muscles. As the bronchi become smaller, when the bronchi are found inside the lung and they divide to become smaller and smaller, the cartilages in the wall of the bronchi become in the form of plates, and then these plates will start to become smaller in size. The outermost layer is an adventitia, and this is a connective tissue layer that binds bronchitis around. So this image is a cross-section through a bronchus. This is the epithelium, and as you can see, it is folded due to the presence of muscles. And here we have the smooth muscles scattered along the wall. Now, because the smooth muscles in the bronchi are helically arranged, they spiral around the bronchus, we will not see the smooth muscles completely surrounding the bronchus in a section, but we'll see them as strands in different regions of the wall. Also, we can see the area where the glands are present. This is the submucosa. And here we can see the plates of cartilage. This is a plate of cartilage. This is another plate of cartilage. And here also we have another plate of cartilage. Now, since we can see plates of cartilage, this means that this is one of the smaller bronchi that are found inside the lungs. If the cartilage was a ring completely surrounding the wall, this means that the bronchus is one of the larger ones that are found outside the lung. Now, bronchioles. Bronchioles arise from the smallest of the bronchi. When do we say that this is a bronchiole? A bronchiole is that part of the bronchial tree that is characterized by the following features. The diameter is very small, usually less than 1 millimeter. Absence of cartilage. This is very important. There are no cartilages in the wall of the bronchioles. Once cartilages disappear, we say that this is a bronchiole. No glands. Absence of glands. There are no glands in the wall of the bronchioles. Smooth muscles become more. Smooth muscles become more. Again, in the trachea, we had smooth muscles on the posterior aspect of the trachea. In the bronchi, the smooth muscles were formed in the form of a spiral around the bronchus, but here in the bronchioles, they form a complete circle. They form a complete circle around the wall, the smooth muscles. If we look at this image here, this is the epithelium. Again, it is folded due to the presence of muscle, and all this is muscle. All this is muscle. Okay. Now, as you can see, almost the entire wall of the bronchiole is smooth muscle. Now, don't get confused. The wall of bronchiole is thinner than the wall of the trachea and the bronchi, of course, but the proportion of smooth muscle is more. The proportion of smooth muscles is more. Of course, on the outside, we have adventitia, connective tissue adventitia. Now, the part of the lung aerated by a bronchiole is called the pulmonary lobule. So all the region of the lung into which a bronchiole conducts air, this is called pulmonary lobule. It is pyramidal in shape and it is surrounded by connective tissue. Now, what about the epithelium of bronchioles? The larger bronchioles are still lined by respiratory epithelium. But we have fewer goblet cells. As the bronchioles become smaller, the epithelium will change and it will become simple columnar ciliated epithelium. Simple columnar ciliated epithelium. And in the smallest of these bronchioles, which is the terminal bronchiole, the epithelium will be simple cuboidal ciliated epithelium. So simple cuboidal ciliated. Importantly, we have no goblet cells. No goblet cells. So here we do not have goblet cells, and from the previous slide we said we have no glands in bronchioles. So what makes the secretions in bronchioles? We'll talk about that in the next slide. Brush cells and small granule cells are still found at this level, and the lamina propria contains elastic fibers. So the wall of bronchiole is simply a mucosa, a smooth muscle layer, and an adventitia. Simple as that. Mucosa, smooth muscle layer, and adventitia. Now we have a very important cell in the respiratory system, which is called Clara cells. Clara cells start to appear in the simple cuboidal epithelium of the terminal bronchioles. These are found starting from the terminal bronchioles. These cells are non-ciliated, they are dome-shaped, and the cytoplasm contains rough endoplasmic reticulum and Golgi apparatus, numerous cisternae of smooth endoplasmic reticulum, and numerous secretory vesicles. Of course, because they have rough endoplasmic reticulum and Golgis, they synthesize proteins. They have smooth endoplasmic reticulum. They could be involved in detoxification, and they have secretory vesicles, so they secrete various substances. And actually, this is the function of Clara cells. Secretions of surfactant and antimicrobial agents, detoxification of harmful substances, and they may even act as stem cells. So these are all important functions for Clara cells. Look at this diagram here. This is Clara cell. This is Clara cell. Rough endoplasmic reticulum, Golgi apparatus, and here we have secretory vesicles, produces the secretion to the outside. In this view here, this is Clara cell. The view on the left, we have Clara cells. The small dots here, the small dark dots, these are the secretory vesicles of Clara cells. Now, respiratory bronchioles. Now, this is the first part of the respiratory zone. We have completed the conducting zone of the respiratory system, and the respiratory bronchiole, these are the first parts of the respiratory zone. They branch from terminal bronchioles. The wall of the respiratory bronchioles has the same histological features as that of the terminal bronchioles. So it's simply mucosa with smooth muscles and adventitia, and the mucosa is simply nothing more than epithelium plus lamina propria. So what makes this different from the terminal bronchioles? The respiratory bronchioles differ from the terminal bronchioles in that alveoli open along the wall of the respiratory bronchiole. There are openings of alveoli along the wall. And this is why respiratory bronchioles are considered the first part of the respiratory zone, because gas exchange will start to occur in this area. Clara cells are more abundant, and they become even more abundant as we go down the respiratory bronchioles. Now, distally, the respiratory bronchioles will give rise to alveolar ducts, and these are tube-like structures that are completely lined by alveoli. So along the entire wall of the alveolar ducts, we have openings of alveoli. The epithelium of the ducts and the alveoli is simple squamous epithelium, nothing more than simple squamous epithelium. Smooth muscles, however, may be present, but they are restricted to the alveolar openings, only the regions where the alveoli open into the ducts. The thin lamina propria is rich in elastic fibers and capillary networks. So now, again, the mucosa is formed of epithelium and lamina propria, and the lamina propria has many elastic fibers. Alveolar ducts terminate in alveolar sacs. These are, from the name, they are spaces, and these spaces are surrounded by alveoli, and these alveoli open into these sacs. But also sacs may directly open along the wall of the duct, so not necessarily from the termination. So in this image here we have a terminal bronchiole that give rise to two respiratory bronchioles, and from this respiratory bronchiole we have an alveolar duct. This alveolar duct ends in an alveolar sac, and into the sac several alveoli open. And here in this image, this is an alveolar sac, and into the sac several alveoli open. So these two images represents the respiratory zone of the respiratory system. Now, what are alveoli? Alveoli are sac-like evaginations, so they are outpouching of the respiratory bronchioles, alveolar ducts, and alveolar sacs. So they are just something that comes out of the mucosa of these structures, the respiratory bronchioles, the alveolar ducts, and the alveolar sacs. They are the site of gas exchange between air and blood. But there are about 200 million alveoli in each lung, with a total area of 75 meters square. So imagine the entire alveoli forms an area of 75 meters square. This is why the alveoli and the airways, or the other airway passages that we talked about, give the lung a spongy structure. Anything that is filled with air, almost entirely filled with air, with thin septa between the air, this is called spongy-like structure. Alveoli are separated from each other by connective tissue called the interalveolar septum. However, alveolar pores are found between the alveoli, passing through these septa, and they connect the alveoli together. This is important to equalize the pressure in the alveoli and to create collateral passages in case of obstruction. So here we have an alveolus, and here is another alveolus. These are all alveoli, separated from each other by septa formed of connective tissue. And here we have these pores. These are the alveolar pores. So notice how this alveolus opens into this alveolus through this pore, This pore will make sure that the pressure in the alveolus number 1 is the same as pressure in alveolus number 2. And in case alveolus number 2 is blocked, air will still pass through alveolus number 1 and will pass from alveolus number 1 to alveolus number 2 through this pore. Now, the interalveolar septum is formed or contains fibroblasts and macrophages. The extracellular matrix is sparse, not too much, but very importantly, we have elastic and reticular fibers. Now, expansion of the alveoli during inspiration is an active process. So we have contraction of muscles that lead to the movement of the air from the outside to the inside of the lung. This will cause expansion of the alveoli. The elastic properties of the alveoli, because the alveoli are surrounded by elastic fibers, we will have a passive recoil of the alveoli, so they will return back to their normal size after being expanded, and this will help push the air to the outside again in the process of expiration. Reticular fibers are also important because they prevent collapse of the lung and the overdistension of the alveoli. So when the alveoli expand, what prevents them from falling apart? It is the reticular fibers, because they form a network around the alveoli that hold the alveoli together, so they prevent the overdistension. And when we have expiration, we have a negative pressure inside the alveoli. So what prevents the collapse of the alveoli? Also the reticular fibers that make the alveoli held together, that hold the alveoli together. Capillaries are also found in the interalveolar septum. Capillaries are continuous, they are of continuous types. And most of the endothelial organelles are located around the nucleus. The rest of the cell is just cytoplasm. It is very thin, and thus it is suitable for gas exchange. What about the alveolus itself? What are the cells that are found in the alveoli? We have type 1 alveolar cells, also called type 1 pneumocytes. These are thin squamous cells. These are the simple squamous epithelium that line the alveoli. Most of the lining of the alveoli are type 1 alveolar cells. Again, most of the organelles are found around the nucleus, and the rest of the cell is thin cytoplasm, suitable for gas exchange. The various type 1 pneumocytes are connected to each other by desmosomes and tight junctions. Type 2 alveolar cells, or type 2 pneumocytes, also called septal cells, these are cuboidal cells that bulge into the cavity of the alveolus. Now they are connected with each other and to type 1 pneumocytes by desmosomes and tight junctions. The nucleus is round and the cytoplasm is filled with vesicles. Now, when we examine the vesicles under the electron microscope, we will see that they are laminar bodies, which means that they contain several layers, as if they contain several layers, like this. These are specific for these cells. These bodies are specific for these cells. Type 2 cells produce surfactant factor. This is important, and this reduces the surface tension and prevents collapse of the alveolus. And also, they may act as stem cells. These cells may act as stem cells. So these cells, the type 2 cells, the type 2 pneumocytes or septal cells, are similar to Clara cells in both shape and function. Alveolar macrophages, also called dust cells, these are present in the alveoli and in the interalveolar septum, as we have said earlier. They are macrophages, so they phagocytose, they engulf foreign particles, they engulf microorganisms, and they engulf dead red cells. Now, as we have talked about in your previous year, and as you are going to take in other areas, this is one of the normal functions of macrophages, to engulf red blood cells, because dead or old red blood cells must be destroyed, and this happens in the liver and the spleen. Now, usually, the macrophages will digest the contents of the RBCs. And of course, as you know, RBCs contain hemoglobin, and macrophages will digest this hemoglobin, will metabolize this hemoglobin. One of the products of metabolism of hemoglobin is a substance called hemosiderin. This is actually a brownish dye. It is brownish in color. So accumulation of hemosiderin inside the macrophages will stain this cell brown in color. And if this happens, this macrophage is now called a siderophage. As you can see here in this region, this is a macrophage that is brown in color. This means that this macrophage has engulfed an RBCs and digested it, and cytoplasm is filled with hemosiderin. As I have said, this could be normal, for example, the spleen or in the liver or even in the lung. However, in certain conditions, like congestive heart failure or pulmonary edema, there will be an increased pressure in the alveolar capillaries. The pressure inside the capillaries in the interalveolar septum will be great, and this will push the RBCs outside the blood vessels. These extravasated RBCs are taken up by the macrophages and digested. And therefore, in these conditions, in these two pathological conditions, we have increase in siderophages inside the lung. And finally, we'll talk about the respiratory membrane. Gas exchange occurs through a series of structures collectively called the respiratory membrane or the blood-air barrier. This barrier is very thin. So if I have here an alveolus, and here we have a capillary, and gas wants to pass from one area to another, what are the structures through which the gas must pass through? So first off, we have the alveolar epithelium. Here we have type 1 pneumocyte. The alveolar epithelium, specifically type 1 pneumocyte. And then we have the fused basal lamellae of the alveolar and capillary endothelium. The alveolar epithelium and the capillary endothelium. And then the final structure that the gas must pass through is the endothelium of the capillaries. The endothelium of the capillaries. So these structures are what form the respiratory membrane. Type 1 alveolar cells, the fused basal lamellae of the alveolar epithelium and capillary endothelium, and the endothelial cells of the capillaries. This is all about the histology of the respiratory system. I wish you good luck and be safe. Thank you.